my name is Joanne Rubacaba,
And my name is James Warrentor.
and we'll be presenting on bacterial isolate identification. So for this study, the purpose was to identify antibiotic-resistant bacteria from our acquired soil products. Microbial resistance is caused by the overuse of antibiotics. The importance of the study was to find non-resistant bacteria to allow for better defenses against harmful diseases. Our soil came from James's front yard, which was frequently watered with the water service line that is connected to the municipal water supply in the city, which includes canals, ditches, and pipes. So it is very possible that there might be a vast amount of bacteria present. So due to this, we planned on seeing our soil producing a great yield of bacteria that has antibiotic resistance. So for antibiotic production, our bacteria showed that there was ability for antibiotic resistance. The antibiotic resistance was seen only on the S10 Streptococcus antibiotic disc. However, it was not shown on the CC10 or CC2 clindamycin or B10 bacitracin disc. As you can see on the right image, there's only clearing on the S10 disc, where the other two, there was no clearing.
Okay, and moving on to the gram stain analysis, we concluded that our gram stain was gram stain positive, meaning our bacteria had a peptidoglycan cell wall. And as you can see in the images that we took here, it had the shapes of a cocci and spherical shape when we tested the gram negative test, and then for the gram positive test, it ended up ended up being a rod shape and also in a pink color form. So for our biochemical test, on the acid tolerance test, it did show that there was still growth. So our bacteria did end up being resistant to the acid tolerance. And for the oxidase test, it showed that it was positive. For the catalase test, it was also positive. And for the citrate utilization, the bacteria was positive with the blue pigment in the tube. Now for the testing of O2, it showed that there is aerobes or bubbles producing, meaning that oxygen was required for growth of our bacteria. For the phospholipid hydrolysis, we also got a negative result for this. And for the blood agar, we did end up getting some gamma hydrolysis that was present. And for a string test, it didn't pass the string test, meaning that our soil was not gram negative, but gram positive instead.
So in conclusion, our test results show that our bacteria was possibly Staphylococcus epidermis. Staphylococcus is a gram-positive bacteria belonging to over 40 species. It causes wound infections, boils, and oozing blisters. Staph can also result in food poisoning with such symptoms of nausea, vomiting, diarrhea, dehydration, and low blood pressure. In rare cases, the bacteria can turn deadly if the bacteria invades the blood deeply or enters the bloodstream, resulting in fever or joint muscle or joint pain or joint muscle. This could be seen since our morphology was cocci shaped or spherical shaped. Our catalyzed reaction was positive. Our lactose fermentation developed a positive reaction, and our mannitol fermentation, which was not included in this presentation, was negative. So thank you for watching this presentation, and I hope you guys have a great summer break.
All right, thank you.